Thank you, Sharon, and thank you to the NHGRI for organizing this. It's been an absolutely fascinating and I think a remarkable couple of days. I come from NCI, the 800-pound gorilla in the NIH system, by far the largest institute with a very large commitment to genomics. And the more recent has been a signature event, the Cancer Genome Atlas, done in conjunction and close collaboration with NHGRI. And we have continued on in that space. And I sort of, as we listen to these discussions, I wanted to encourage all to at least consider looking at many of these data resources, because cancer, the devolution, not the evolution, but the devolution of cancer is really nature's errors that are showing us all sorts of remarkable things about how certain pathways and interruptions or disruptions of particular genes have downstream effects, as well as what are the upstream effects of those. And so I think it's really quite a remarkable opportunity, and want to encourage any and all to look and think at these data sets in novel and different ways. And I think that's very important. So we have a very large portfolio. We have about 600 studies in and around dbGaP, the old SRA and the like, and we are moving towards a large cloud type of solution. The approach, I think, for the NCI for many of these large studies is going to be bring the analysis to the data, because so many of the data sets are so large. TCGA with 12,000 fully characterized genomes, and you'll see in a second we're doing another 15,000 over the course of the next 2 to 3 years. These are enormous data sets. It's just very difficult to move around, and so the idea of bringing transparently the analysis to the data is a critical, I think, sort of shift in the way of thinking about these things. As you can see here, we're doing a lot of clinical sequencing, sort of the clinical trial, or sorry, that's misspelled there, in a number of different cancers. Targeted therapy is very much a very exciting thing, as is immunotherapy, and how to understand how the responses can indeed be predicted so that we can be much more precise in our decisions as to what to give and when to give it. But I highlight one thing I think of particular interest that Loosed Out and others have started with Mike Stratton. And this is human cancer model initiative that's going to target roughly 1,000 organoids that are developed, that are going to be available through commercial outlets, where they'll be sequencing at a certain point of the development of those, and then those resources are available for people to go CRISPR, knock in, knock out, do whatever they want. But there'll be up to 1,000 of them. The first 150 are in place already. And I think this is a resource for those who are thinking about different tissue origins and the like, really should be going and looking at that. And so we're very excited about that. I think the other thing is we have a very strong commitment to molecular epidemiology. In our program, we're leading a large genome-wide association study of 350,000 breast cancers. About 150,000 have been scanned. We have the resources, and we're building, again, a cloud-based solution for this for the next 200,000 plus controls, going multi-ethnically is a very strong component of this, and the controls for this. So we think this is very important, with a number of environmental exposures that have come from epidemiologically rigorous case control and cohort studies. Similar things are being contemplated for prostate and colon cancer. So I think it's really important to be thinking about these very large data sets, where we have some interesting questions of disease heterogeneity versus disease homogeneity that I think are really important methodologically as well as scientifically, in giving us very clear ideas. So with that, I'm going to transition now, take off my cancer hat. Even though I come from that cancer perspective, the first thing that I want to talk about may seem like cancer, but in studying cancer, it turns out it's not cancer. And that really is this question of genetic mosaicism. It's really, in the early 210, 212 range, we started to see these in GWAS, and the sequencers were seeing the same thing, that there's subpopulations of cells. And the question is really, what is the extent to which we see this tissue by tissue, age specific per se, and then what does this perhaps portend for many of the different chronic diseases and or responses to therapy? This is very much of an open question. We're just beginning to see this, and so this is sort of filtered through some of the conversations, but I wanted to put out there possibility of doing a very deep catalog of events across many different tissues, really asking at different ages, what does this look like? Even considering, the NICHD project in placenta, what does it look like from the very earliest point to the much older? We know, unfortunately, about 40% of the men in this room over age 50 have loss of Y chromosome in some fraction of their circulating hematopoietic cells. What that means, there's controversy over the specifics of how predictive that is, but these are events that are going on. Our genome is falling apart. And how to understand this at the level for tissues of specific single cell versus colonies, so to speak. There have been some really exciting papers in Science and Nature recently looking at this in esophagus, in skin, in lung areas of the body we know that have continual environmental bombardment, and seeing just the degree of post-zygotic somatic mutations that are there and very different rates and different distributions of those mutations that don't all necessarily go on to develop cancer raises this very interesting question of that yin and yang between the generation of mutations and how well or not so well we repair those things. And with age, I'm afraid that is kind of the definition of age in my mind, is the less capable repair per se. So the question is how to do this in a large way, in a sufficiently powered way. I mean, GTEx is a great start, but we really need, I think, something considerably larger where you would also have the opportunity to have organoids, et cetera, generated from some subset for very important mechanistic laboratory-based evaluation. Along with imaging analysis and artificial intelligence, which is really revolutionizing imaging, and particularly the nature of being able to have mechanistic as well as predictive outcomes associated with both radiographic and histologic activities. Warm autopsy protocols that are being contemplated, particularly we have one, we started with BRCA1 carriers who do not die of cancer. And the question is, what are the implications of carrying a BRCA1 mutation in other tissues, lung, muscle, and the like? And I think these are the kinds of things that may be incredibly informative. The possibility of longitudinal study, and I mentioned the placental. The second thing I wanted to raise is, again, the loss of function, particularly both in homozygote and heterozygous state, is one that's come up. And NHGRI has a very rich tradition with the undiagnosed disease program of really taking interesting enigmas and genetically sorting them out. I would take this model and suggest you may want to look at it a little bit differently in identifying individuals that come out of population-based sequencing or any number of cohorts or disease-specific studies where you have 55-year-old men with CFTR double, knockouts, but yet are running marathons. What can we learn from deep phenotyping of those and much more, I think, elegant characterization of the dynamics of their, whether it's their T cells who are looking, at single cell and really going a very deep dive into these really remarkable individuals. The sickle cell, the homozygous sickle cell patient who's 61 years old and not had a stroke. There's an awful lot to be learned on these edges, and I think with all the population sequencing going on, we probably are going to identify more and more of these individuals, and I think there is a real opportunity to try and really characterize in what context. We're doing this in cancer with what we call the Exceptions Project in the NCI, individuals who were 5 or 10 years out with a fatal disease like pancreatic cancer, going back and sequencing their tumor, trying to understand why is it that they've responded to therapy. Why are they the 1 or 2% of pancreatic cancer who are stage 4 who've responded to a given therapy? And so this kind of model, again, looking at the edges, keeps coming up with PRSs and EQTLs and the like. But here, I think we are identifying these individuals and how and in what way can we characterize them. I think that's a very important way to think about this. So why don't I stop there? I think we're going to go through the presentations, right?
Yes, and so Howard Chang from Stanford's going to talk next. I would just say about the somatic mutation, it didn't get brought up at this meeting nearly as much as our panel thought it would, so we wanted to put it back out there, even though it didn't get discussed, because clinically we think it's very important.